Hi everyone, this is Bhagilakshmi, Department of Biotechnology, KSRCT. In this video, we are going to discuss about single cell protein. What is single cell protein? How they are manufactured? When we have started using this single cell protein? And what are the microorganisms involved in the production process? And what are the biotechnology process involved in the production and purification? And what about the composition of the SCP? And its advantages and disadvantages, and about some most important part is its application. What are single cell proteins? The term single cell protein refers to the total protein which is extracted from the pure cultures of microorganisms, example yeast, algae, filamentous fungi and bacteria, and this can be used as a protein supplement for human and animals. And this is used as an animal feed and can be used as a human feed as a protein supplement, and it is also called as novel food and mini food. About the history, this has been a part of our diet since ancient time, earlier known as microbial protein. The name was first introduced by Professor Scrimshaw of MIT in 1967. The transportation of food was very common in past, but with the increasing in population, energy crisis has encountered throughout the world. So they have started thinking about alternate food that should be of rich in protein to overcome this energy crisis. In 1960s, the idea of using dried cells of microorganisms which can be an ultimate part to solve this problem. Thus, more research work has been carried out among scientists in this area, and industries have started producing this single cell protein in large quantity. In 1950s, British Petroleum initiated production of SCP in commercial basis. In 20th century only, the SCP technology for the production of protein-rich contents from the microbes on the large scale was established. In 1960s, British petroleum industry developed a technique named as protein from oil process using yeast by feeding on wax paraffin, a product of oil refinery. Coming to the microorganisms used in the production of SCP, we have yeast. This comes first. In the World War I, one of its species, Candida utilis, other name for us, torula yeast, was produced by Germany and it is used in soup and sausages commonly. And this is also used commercially as seasoning of food, and one of the example of this is hickory smoked dried torula yeast. The baker's yeast, which is known as Saccharomyces cerevisiae, is used commercially in food bakers. Suitable substrates for this are molasses, cornstarch, liquor, etc. under the controlled conditions like temperature 25 to 26 degrees Celsius, and aeration should be there. pH must be of 4.4 to 4.4, and carbon source, nitrogen source and sulfur source has to be provided. And yeast have many advantages of using in the production of SCP when compared with other microbial group, other sources also. This biomass can easily harvested because it has a larger size when compared with other groups. And also it has a long traditional history and its usages. And this produces high level of lysine and malic acid. And also it can grow at high acidic pH, whereas there are some disadvantages have been noted. Yeast have low growth rate when compared with bacteria, and also produces low overall protein content about 45 to 65 degrees Celsius. Coming to the second group, filamentous fungi. In 1973, actinomycetes and some other filamentous fungi have been used to produce SCPs. It requires optimum temperature of about 25 to 30 degrees Celsius and pH 5 to 6 degrees Celsius, 5 to 6 or below, an ammonium salt acting as a nitrogen source. Advantages of using filamentous fungi are It has a faster growth, it has a high protein content, it has a higher penetration power into substrates. Disadvantages are some fungi have very slow in growth and also produces mycotoxin that causes some health issues in human and animal, and also risk of high contamination. So these are acting as a disadvantages of using filamentous fungi. Coming to the next microbial group, algae. So many algae have been used as a mass cultivation, example chlorella and spirulina, etc. They are highly rich in protein and it's very simple and fast growth, requires no carbon dioxide aeration but require temperature 25 degrees Celsius and pH 8 to 10 have to be maintained for biomass. Coming to the bacterial group, it requires pH, optimum pH about 5 to 7 and temperature about 35 to 45 degrees Celsius. Advantages of bacterial strain are having higher growth, rapidly it grows, having shorter generation time of about 2 to 20 minutes. Utilization of any kind of raw material as a carbon source. So these are benefits of bacteria in using production of ACP. So this table will tell you the different composition of microorganisms in percentage dry weight. Coming to the protein content, among all these group, bacteria produces high percentage of dry weight, that is 50 to 65% protein is there in bacteria when compared with other group. Low fat has been observed in bacteria 3 to 7%, and nucleic acid content is more in bacteria but very less in algae. So these are the microorganism list used for the SCP production. In fungi, Aspergillus fumigatus, Aspergillus niger, Rhizobus cyclopeum has been widely used in SCP production. In yeast, Saccharomyces cerevisiae, Candida tropicalis, Candida utilis has been used. Algae, Spirulina species, Chlorella pyrenoidosa, Chondrus crispus, Bacteria, Pseudomonas fluorescens, Lactobacillus, Bacillus megaterium. So these three bacterial genus have been widely used in the production of ACP. Coming to the comparison of microorganisms widely used in SCP production. In fungi, it is easy to grow fungi and harvest, but very low growth rate and low protein content. Coming to algae, easy to grow and harvest, high quality protein is there in algae, but due to the non-digestible cellulosic cell wall, acting as a high disadvantage, and also it will concentrate heavy metals. Coming to the yeast, it is having a larger size but lower nucleic acid content. Familiarity and acceptability will be there, but disadvantages are poor digestibility in nature, low protein content have been recorded, and very slow growth rate. Coming to the bacterial group, this is having high protein content, having digestible cell wall, but disadvantages are high nucleic acid content, small in size, and having very low density. So in this picture you can see the production of algal biomass. This will grow autotrophically, requires low density of light and requires 35 to 40 degrees Celsius temperature, pH 8.5 to 10.5, and this has been cultivated in large trenches of sewage oxidation pond. You can see green color algal mat. This can be acting as a biomass, and from that SCP has been produced. In this picture you can see the cultivation of bacterial and fungal biomass. This will be grown very easily and wide range of substrates. They require minimum temperature of 15 to 34 degrees Celsius and pH of 5 to 7 degrees 7. And this is yeast biomass cultivation. By using agro-industrial waste such as molasses, starchy materials, fruit pulp, wood pulp, it has been grown. It requires the temperature of 34 degrees Celsius, pH of 3.5 to 4.5. Also requires addition of inorganic acid and sulfur supplement in the form of salt. And these are some factors which influence the growth of biomass production, that is e- elimination time, temperature, pH, suitable strains, agitation, sterile conditions. These are the factors which affects the biomass production. In production of SCP, the following steps have been framed on a selection of strain of microbes and substrate. Second, fermentation. Third, harvesting. Fourth one, post-harvest treatment, processing of SCP. Selection of strain of microbe and substrate is the foremost important step in production of SCP. So the microbe selection should not produce any kind of toxicity in biomass. It should not harmful to consumer. Selected microbes should produce a large quantity of protein, and also the substrate which is used to produce cultivate microorganisms should be cheap, effective, allow favorable growth, and ease in isolation. Coming to the second step called fermentation process, it is done in large chamber either in glass or stainless steel called fermenter. This fermenter will have all controlled conditions, produce sterilized conditions such as optimum temperature, optimum pressure, pH, humidity provided by the fermenter. Usually fed batch cultures have been used in the production of SCP by fermentation process. When third step, harvesting, when colonies have been grown fully developed, then they are harvested. They are removed from the fermenter by decantation process. The next step, post harvesting fermentation. After microbial colonies have been harvested, the cells were subjected to various process treatments includes separation by centrifugation, washing and drying, etc. So this is called post harvesting method. Coming to the processing step, the produced protein which may contain impurities, for example carbohydrate, nucleic acids, lipid content and some kind of salts, that has to be removed from the protein. Pure protein isolation will be done by disrupting cell wall through crushing, crumbling, cycling of freezing, thawing, grinding and thermal shocks. So these are the methods have been applied to extract protein from microbial cells. And also nucleic acid has to be removed from the protein by treating with 10% of NaCl, by adding sodium hydroxide chemical and by going for thermal shock, by using enzyme treatments such as ribonuclease enzyme. These are the advantages of single cell protein. Microbes which are having a rapid generation time at a particular period, they can grow for many generations. Among all microorganisms, algae is having 2 to 6 hours of generation period, yeast 1 to 3 hours. Bacteria is having very less generation period, 0.5 to 2 hours. So they can be easily modified genetically for better amino acid composition in protein. They contain 43 to 85% of protein, their dry mass. This can be used variety of raw materials as the source of carbon and thus can use the removal of pollutants from the environment. High yield and good composition of SCP has been observed, can all be produced isolated easily and it is beneficial ecologically. Coming to the disadvantages of SCP, some microbial groups produces toxin that may be harmful for both human and animals, causing diseases in both groups. So that sometimes leads to allergic reactions and indigestion problem. Higher nucleic acid content of SCP lead to human kidney stone, and this is the production process involves very expensive and also require high sterile conditions. taste may change and some unacceptable coloration produced in this SCP. Coming to the application part of SCP, this SCP acts as a protein supplemented food. It acts as a source of vitamin, amino acid, mineral, crude fiber, etc. Supplemented food for undernourished children. Directly, this can be taken by them and they can improve their protein strength. It acts as a health food. It controls obesity. It provides instant energy. For example, spirulina acts as a part of diet of US Olympic team. This SCP can also act as a therapeutic and natural medicines to reduce body weight, cholesterol, stress. It lowers the blood sugar level in diabetic patients due to the presence of beta linolenic acid. It prevents accumulation of cholesterol in body and also improve your skin and eyes presence of beta carotene in SCP. This beta carotene acting as anti-cancer substances which has been identified by UN National Cancer Research Institute. And also this SCP increase lactation. This SCP also used in cosmetic industry. It play important role in maintaining healthy hair by incorporating vitamin A and B. Many herbal beauty products have been released. Bio lipsticks and herbal face cream have been produced capable of replacing coal tar dye based cosmetics. And also this SCP acting as excellent convenient source protein and other nutrients. It is used to feed cattle and fishes, etc. Coming to the conclusion of SCP, so this development of single protein, it's just a beginning of biotechnology. So with the improvement and development of techniques in biotechnology will improvise or solve the malnutritive conditions of the progressing countries and can also introduce better quality of food and taste with the decreased chances of occurrence of side effects. This will be possible only with the biotechnology. Moreover, genetic modification in microorganisms can also lead a better future of SCPs in biotechnology, medicines, agriculture and poultry, etc. Thank you for patience listening.